Eric Nestler is our first speaker. Eric Nestler is a New Yorker. He's the Nash Family Professor of Neuroscience at Mount Sinai School of Medicine. At Mount Sinai, he's the Dean for Academic and Scientific Affairs, and he's the director of the Friedman Brain Institute. So Eric started out his career at Yale, where he did his undergraduate studies and then went to medical school. And then he stayed at Yale as a professor between 1987 and 2000, so a good long time. In the year 2000, he was recruited to UT Southwestern in Dallas, where he became chairman of Psychiatry. And he stayed at UT Southwestern until 2008, when he moved to New York to Mount Sinai School of Medicine. Everybody probably knows or is in some degree familiar with Eric Nestler's research. His focus is on depression and on addiction, and he really studies the gene expression, the gene expression that is associated with depression. He's going to give us a marvelous story today on sex differences in the molecular basis of depression, and be a role model for a modern psychiatric scientist. With that, I'll invite Eric Nestler to come up.
Thanks so much, Carol, and thank you for inviting me to speak here this morning. Welcome everybody to New York City. I was asked to begin with a slide of disclosures, so I'll just give you a second to take a look at those. And what I'll be talking about today is two aspects of depression. First, I want to focus on dramatic sex differences that are seen in depression. We've known for many years that depression is twice as common in women than men, but the molecular basis of that sexual dimorphism has remained incompletely understood. And second, I want to talk about the molecular mechanisms by which early life experience programs an individual's susceptibility to stress for a lifetime, with, again, very prominent sex differences seen in those underlying mechanisms. And in both cases, what I'll do is to illustrate the ways in which we've used large-scale, genome-wide assays to gain insight into both of these questions, focusing on transcriptional and epigenetic mechanisms. And I'll begin by giving you a very brief description of what I mean by transcription and epigenetics. So we know that a mammalian organism, a mouse or a human, has about 3 billion nucleotides in their genome. If stretched out linearly, that DNA would be about 2 meters in length. And somehow we've learned that this 2 meters of linear DNA is compacted into a microscopic cell nucleus. Truly, I think, one of the amazing solutions developed through evolution. And we've learned a lot about how that DNA is compacted in a nucleus. I don't think I have a laser pointer, but I think you could follow along with here. That the DNA double helix is wrapped around octamers of histone proteins to form the unit of chromatin called a nucleosome. Nucleosomes are then further organized in very extensive ways, and in the most compact chromatin, excuse me for 1 minute. That's why Carol is president. Thank you, Carol. Good, so the nucleosomes are further compacted in the most compact state to what we recognize as a chromosome. This organization is very important functionally because linear spans of DNA that are in compacted regions of chromatin is the material inside of a cell nucleus. For example, DNA contained here is functionally inactive because it is simply too compacted for its linear sequence to be read. In contrast, DNA that is in the more open regions of chromatin can be active and encode RNA and play other regulatory roles. We can take advantage of this information in several ways. We can use the information to help us identify those specific genes that are affected in human depression or affected in mouse models. Importantly, from a mechanistic point of view, studies of chromatin have provided the first ever look into transcriptional mechanisms that occur in brain cells in vivo. And by analogy with other fields, for example, developmental biology and cancer biology, where certain types of changes in the compaction of chromatin, so-called epigenetic mechanisms, once they occur, are permanent, the hypothesis is that perhaps a similar type of permanent epigenetic change occurs in certain cells in the brain that lead to long-lasting changes in an individual's behavior in response to life experiences. Let me tell you a little bit more about these mechanisms, focusing on this level of analysis. Now the DNA are these gold stripes wrapped around these spheres, the histone octamers, showing chromatin in the two extremes, one open, active, one closed and inactive. In the active state, there are open linear sequences of DNA that can be bound by sequence-specific binding proteins called transcription factors. Delta Phos B and Creb are just two factors that my lab has studied. There are literally thousands. The binding of transcription factors to their active binding sites recruits many proteins to the vicinity of that gene to open the chromatin further, leading to gene transcription. Some of these mechanisms of gene activation involve modifications of the histone proteins, for example, as indicated by these letters, acetylation, phosphorylation, methylation of histones, and so on. At the opposite end of the spectrum, mechanisms of gene repression maintain chromatin in a very compacted state, also mediated by changes in histone proteins, changes in DNA itself, and many other mechanisms. All told, in simple systems like yeast cells or stem cells, where this is possible to study, it has been demonstrated that the opening or closing of DNA around a specific gene, mediating that gene's activation or repression in response to an extracellular stimulus, can involve 200 proteins at a single locus in the genome. So these are extremely complicated mechanisms. Starting about 10 years ago, when we moved back to New York, my lab reoriented itself, and rather than focusing on individual genes or proteins that we thought might be important, we decided to take a completely unbiased, open-ended approach. I think this is really important because we don't know enough yet about the brain or its diseases to always guess right into what gene or protein to study. So that approach is depicted here in cartoon fashion. We would use next-generation sequencing methods, for example, RNA sequencing, to capture all the RNAs expressed in a tissue or cell type in a certain state, in a chronic stress model or in human depression. We would capture RNAs that are upregulated, for example, and others that are downregulated. And if those RNAs are regulated through transcriptional mechanisms, changes in that RNA would be associated with changes in chromatin at the vicinity of that RNA's encoding gene. And we would capture those chromatin changes through related methods called chip sequencing and many other types of experimental approaches. So, for example, we would identify changes in histone proteins or DNA that would an increase that would reflect an increased transcription and opening of chromatin, or in the opposite, a reduction in some repressive mechanism. And we would overlay on top of those data still additional data sets, chip sequencing, for the key transcription factors that we and other labs have believed are important in mediating some of these responses. This is a tremendous bioinformatic challenge because the sequencing data illustrated by this slide, one brain region, one time point, one experimental model, would involve perhaps 20 terabytes of sequencing data. So we're still very much in the process of figuring out how to optimize the mining of these data sets, and I'll give you some progress reports today of how we're doing that. So the first data set that I'll mention briefly is work done by Rose Baggett in the lab. Rose is now at McGill. And what Rose did was to employ a chronic stress model in mice that my lab and others have used, chronic social defeat stress. Mice are subjected to social stress for 10 days. Rose analyzed these mice 2 days later. She analyzed 4 brain regions by RNA sequencing: nucleus accumbens, medial prefrontal cortex, basolateral amygdala, and ventral hippocampus. What I'm showing you are genes that are affected in resilient mice and how those same genes are affected in susceptible mice. Yellow means the gene is induced, blue means the gene is repressed. Susceptible and resilience refer to the response of individual mice to the same 10 days of social stress. About 2/3 of C57 genetically inbred mice show a full range of behavioral abnormalities. We call them susceptible. The remaining third avoid most of those behavioral abnormalities, even though they're subjected to the same degree of stress. We call them resilient. The Venn diagrams at the right show the exact same data, simply comparing the total numbers of genes up or down regulated in the resilient state versus the susceptible state. And just to illustrate how we've used these large data sets, how they can help us formulate hypotheses, is that we had initially hypothesized that resilience reflects the lack of bad things that occur in susceptible mice, right? That would be one commonsensical hypothesis. And that does occur to some extent, but by far the predominant pattern is different. The predominant pattern is that many more gene expression changes are seen in most brain areas in resilient animals, not susceptible animals, suggesting that resilience is the more plastic state and that animals that are susceptible might succumb because they lack that inherent adaptive plasticity, raising the novel possibility of developing new approaches to treat stress-related disorders, not by undoing the bad effects of stress, but also by reconstituting mechanisms of natural resilience. Now, there have been some increasing concerns about the use of animal models in psychiatric disorders, and I'm going to come back to that in just a moment. But because of those concerns, my lab and other labs have worked increasingly to validate all findings from animal models in human postmortem brain tissue. So recently, Benoit Labonté in the lab, he's back at Laval University in Quebec, carried out a large RNA sequencing study in human depression on 100 humans, half depressed, half not depressed, half men, half women, from 2 brain banks, one in Dallas with Carol Taminga, and the other from McGill with Gustavo Turecki. And what I'm showing you here are RNA sequencing results from 6 brain areas, including ventral medial prefrontal cortex, Brodmann area 25, several other regions, and the nucleus accumbens included as well. And one of the dramatic findings in this data set were differences in gene expression abnormalities seen in depressed men versus those seen in depressed women, differences which are highlighted in the Venn diagrams to the right. We were struck by this dramatic lack of overlap, only 5 to 10% concordance in these gene expression abnormalities. And I'm delighted to say that Marianne Senny's group at University of Pittsburgh just published a study on a completely separate cohort of depressed humans, validating this finding of virtually little overlap in the depressed state between men and women. This difference in gene expression abnormalities in depressed men versus depressed women is recapitulated in chronic stress models in mice. So we've seen by chronic social defeat stress, as summarized here, but also with another chronic stress paradigm, which is called chronic variable stress, a similar lack of overlap in gene expression abnormalities induced by the chronic stress in male mice versus female mice. And I'm showing you the data from one of those chronic stress paradigms. The important thing about this paradigm is that after 21 days of chronic variable stress, Georgia Hodis in Scott Russo's lab with Benoit Labonté has shown that male and female mice display equivalent degrees of behavioral abnormalities, both qualitatively and quantitatively similar behavioral outcomes to the stress. Yet despite that confluence in behavior, the gene expression abnormalities that chronic stress has induced are largely non-overlapping. Now, the overlap is a bit more than what's seen in human depression, but let me emphasize this experiment. Genetically inbred animals subjected to identical levels of stress, where we know that stress induces equivalent behavioral abnormalities, and yet gene expression abnormalities overlap between the sexes by only 10 or 20%. Same is true for chronic social defeat stress and other stress paradigms. So we believe that these data together demonstrate that depression, the broad depression syndrome seen in humans, is a fundamentally different syndrome in men versus women, and it argues for studying the pathophysiology of the disorder as well as the development of new treatments in a sex-specific manner. Now, the availability of these large data sets also enables us to do something quite interesting. So for decades, scientists have debated whether a behavior exhibited by a laboratory animal has face validity for a symptom exhibited by a sick human. That is a complicated question to address. The availability of our RNA-seq data sets allows us to ask more objectively to what extent do different chronic stress paradigms in mice, for example, recapitulate the gene expression abnormalities that are seen in the brains of people with depression, doing this by brain region and by sex. And I'm just showing you an example of this analysis that's ongoing by Benoit Labonte today. And what he's found is depicted in this slide. This slide is not meant to be quantitative, just heuristic. What we have found is that each chronic stress paradigm, and I've already mentioned chronic social defeat stress and chronic variable stress. A third assay that we've used is social isolation of adult animals. Animals raised normally, and when they're young adults, they're housed singly for many weeks. Each of these chronic stress paradigms does successfully induce a subset of gene expression abnormalities seen in depressed humans. You can see the percentages of overlap between the mouse and human tissue in parentheses, but a largely different subset for each chronic stress model, suggesting that each chronic stress model is recapitulating perhaps a different aspect, a different domain of what's seen in human depression. And importantly, this convergence between gene expression abnormalities in chronic stress models in mice with human depression is not seen when we take our mouse data and overlay them with published data sets of other human syndromes, for example, schizophrenia, bipolar disorder, and autism. So as I mentioned, there have been increasing concerns about the use of chronic stress models in mice, but I think those concerns reflect overinterpretation of the models, not the models themselves, and that our data incontrovertibly prove that it is possible to recapitulate broad swaths of the molecular pathophysiology of the broad human syndrome of depression in a laboratory mouse. And it's up to us to then use the data sets accordingly in a way that is consistent with what the models are teaching us. So I'm just going to briefly summarize what we found from our studies thus far looking at sex differences before moving on to the more longitudinal studies of early life experience. We've been able to take some of the genes that show abnormalities only in depressed females or chronically stressed female mice, or other abnormalities only seen in depressed men or chronically stressed male mice, and manipulated those genes in these mouse models, and have been able to show, in fact, sex-specific effects of these genes in stress susceptibility within the animal models. So we believe that this provides further evidence for what I stated earlier of dramatic sex differences in mechanisms of human depression. One of the main interests of the lab and the field at large is to understand what is driving these sex differences. One possibility are chromosomal mechanisms. Another possibility that has been widely studied in the field are known hormonal differences. Differences in life experience are a third possibility, particularly in humans. It's less clear whether the life experience of a developing male and female mouse is different, but it could certainly be as well. Now, an important question to ask is, if human depression is so different between men and women, why is it that in general both sexes respond roughly comparably to available antidepressant treatments? I think there are several ways to understand that. First is, although the overlap in gene expression abnormalities is only 5 to 10% between depressed men and depressed women, that's still many hundreds of genes reflecting considerable common pathophysiology. Secondly, in some of the cases where we've seen very different genes affected in depressed men versus depressed women, the biochemical pathways comprised by those genes is the same. So that a drug aimed at those concordant pathways would be expected to be effective in both sexes. And in fact, most of the mechanisms that my lab and other labs have studied over the years, and I'm just listing a few of them here, are examples of changes that occur prominently in both sexes, both in human depression and in mouse models. But despite that commonality, there are large numbers, thousands of genes, that are affected uniquely in females versus males, arguing for a drug discovery approach that is sex-specific, as I mentioned. For the remainder of the talk, I'm going to shift gears and address a related question of how it is that early life experience programs an individual's own level of stress susceptibility for a lifetime. We know that epidemiological studies have established that early life stress is one of the strongest risk factors for human depression, leading to a depression syndrome that is more complicated and more treatment resistant than in individuals without a history of adverse life events. And there's a large literature in rodents that have studied different forms of early life stress, maternal separation being the most widely used. However, it has been more difficult to identify an animal model of early life stress that produces a latent increase in susceptibility. In other words, more akin to what's seen in humans, most humans exposed to early life stress do just fine throughout life, but remain more susceptible to subsequent stressful life events. We wanted to recapitulate that latent susceptibility in a mouse model. So for the remainder of my talk, I'm going to be discussing how we've addressed this question. This is all work done by Kate Pena in the lab, who's off to Princeton at the end of the year. And what Kate did was to take C57 mice and expose them to a form of maternal separation, reduced bedding material for the moms, for a few hours a day for different periods during the 3 weeks from birth to weaning. So broadly speaking, there was an early life stress 1 and early life stress 2 period. The animals were then group housed again with their litter mates, and then either not exposed to a subsequent stress or exposed to a subsequent stress as adults. And what Kate found is that exposure to that second period of early life stress rendered these male mice, as shown here, more susceptible to adult stress. So that is shown, I'm just showing you 2 behavioral assays. We have many more. In looking at social interaction behavior, under control conditions, the animals are normal, so early life stress had no effect on the baseline behavior of the adult animal. When the animals were exposed to this first period of early life stress, they were still normal, even after a period of subthreshold defeat. But exposure to this second period of early life stress rendered these mice more susceptible, now showing a reduction in social interaction behavior with the 2 hits. This can be depicted in a general resilience susceptibility score, shown in this yellow graph here, showing normal levels of susceptibility in the early life stress 1 period, but enhanced susceptibility after early life stress 2. Changes in the sucrose preference assay provide further evidence for this behavioral outcome. In this paper that was published a year ago, only involved male mice. We have data from female pups as well, who show an equivalent behavioral response. This is taking females, exposing them to early life stress 2 or normal conditions, housing them normally for the rest of their juvenile periods when they're young adults, subjecting them to chronic variable stress, showing that, just as seen with the males, that the females exposed to early life stress are normal behaviorally at baseline when they're adults, but are more susceptible to that second hit of stress. And you can see from this survival plot the dramatic synergy that exists from exposure to these 2 stresses together. To gain insight into the nature of this latent susceptibility, the approach that my lab has used, as I've already depicted, is large-scale RNA sequencing data. So what Kate did was to carry out RNA sequencing on adult animals that had been exposed to early life stress, that second period of early life stress or control conditions, on 3 brain regions of male and female mice. So here we're showing you ventral tegmental area, nucleus accumbens, and ventral medial prefrontal cortex. And the comparison that I'm showing in this slide is comparing the effects of early life stress itself. So this is a single hit early life stress, animals examined in adulthood when they're behaviorally normal, or normally raised animals subjected to adult stress. And what these RNA-seq heat maps reveal is dramatic similarity between the two states, suggesting that one of the ways early life stress might be programming enhanced susceptibility to subsequent stress is by recapitulating some of the gene expression abnormalities that would normally occur in an adult animal after exposure to severe stress. I think you can see the similarities between these heat maps. By the way, I should have mentioned, for those of you who are not used to looking at heat maps like I do every day, each of these vertical lines would reflect a single RNA species. And you can see that there are similar patterns across all brain regions in male and female mice. But interestingly, once again, virtually no overlap between the molecular abnormalities seen in the stressed male mice versus the stressed female mice. Again, recapitulating the general theme, similar behavioral outcomes, but dramatically different underlying molecular mechanisms. So the brain area that we focused the rest of this study on, which I'll show you some additional data for in just a moment, was the male ventral tegmental area. We focused on this brain area because of the following reasons. We were able to demonstrate the strongest statistical similarity in comparing these 2 experimental time points. And a statistical test that one can use rather than just visually saying, well, there's a little yellow here and blue here, is to use what's called rank-rank hypergeometric overlay analysis, which provides a threshold-free way of comparing 2 large data sets, looking at the effect of adult stress by itself and the effect of early life stress with no adult stress. And this bright red color demonstrates that very strong statistical significant overlap between these 2 conditions. And when we drilled into the genes comprising this overlap and asked bioinformatically, of everything that's known about these genes that are regulated under both conditions, what transcription factor would be upstream of these genes? And one transcription factor was the most strongly registered, and that's called OTX2. OTX2 is a transcription factor that's been shown to be important in the development of VTA dopamine neurons, but never before studied in stress models. And interestingly, this predicted role for OTX2 in the male VTA was not seen in the female VTA, nor was it seen in any of the other brain areas that we studied. So we focused this paper only on the VTA and OTX2. So the first thing we did was to drill into our RNA-seq data sets and asked, what is our RNA-seq data showing us about the genes that are predicted to be downstream of OTX2? And what we found is that after a period of early life stress, no adult stress, these target genes are suppressed, even though levels of OTX2 by itself in adulthood are normal. This was very interesting to us. Why are OTX2 target genes altered after early life stress, even though OTX2 is not? So what Kate did was to look earlier in development and found, in fact, that OTX2 levels are suppressed right around the period of early life stress at the mRNA level, but recover to normal during development. That's also seen at the protein level using immunohistochemistry, showing a reduction in levels of OTX2 right after the early life stress, normal in adulthood, and the immunohistochemistry validating that, in fact, virtually all OTX2 in the ventral tegmental area is present in VTA dopamine neurons. Kate then asked whether this early life change in OTX2 is behaviorally significant. So what she did was to use herpes simplex virus vectors. These are prepared for us by Rachel Nevy, a long-time collaborator, now at MGH. These are perfect vectors to use for this experience because HSVs express their transgenes for just a few days. After a few days, the viruses dissipate and gene expression goes back to normal. So what Kate did was to expose animals to early life stress. Immediately toward the end of that stress period, she injected a virus expressing a control protein, for example, GFP, or expressing OTX2, to ask whether this expression of OTX2, just during that brief period of time when OTX2 levels are normally down, rescuing that, what happens behaviorally. And what she found, just looking at the composite yellow graph of susceptibility and resilience, is showing that exposure to early life stress itself induces susceptibility, as I stated earlier, but that overexpressing OTX2 transiently during that time period completely rescues the behavior. And along with rescuing the behavior, we can show that the putative target genes for OTX2 that are all suppressed after early life stress are also rescued for a lifetime, even though the OTX2 rescue was just for a few days earlier in life. Kate could also do the opposite, now using a herpes simplex virus to overexpress a microRNA, a small span, 20 nucleotides of RNA, that suppress levels of expression of OTX2. These animals were not exposed to stress. They were instead simply injected at about that same time period of stress with the viral vector that causes just a few days impairment in OTX2 before OTX2 levels return to normal, or a control vector, showing that simply suppressing levels of OTX2 for a few days early in development can recapitulate that enhanced susceptibility to stress, and it is enough to produce lifelong impairment in the expression levels of some, not all, but some of these putative target genes of OTX2. Kate has more recently examined that ability of a suppression in OTX2 to recapitulate these lifelong changes in gene expression by RNA sequencing, a far better way to do the experiment. You can see here the degree to which simply knocking down OTX2 for a few days early in development in a mouse's life can produce lifelong changes in expression levels of many of the genes that are affected by early life stress. And the overlap is seen in these Venn diagrams to the right. Now, among the target genes for OTX2 identified in these studies are several ion channels, suggesting that perhaps one of the ways in which OTX2 and early life stress affect the VTA for a lifetime is by altering cell excitability, and I'll come back to that in a few minutes. Okay, so how can OTX2 be producing this lifelong impairment in genes even though OTX2 in adulthood is not itself affected? So to examine this question, Kate turned to quantitative CHIP, chromatin immunoprecipitation, where she examined putative target genes of OTX2 early in life or in adulthood. What she found early in life is that under control conditions, each of these predicted target genes of OTX2, looking at predicted sites in those genes that are thought to bind OTX2, indeed bind high levels of OTX2 under normal conditions, and that binding is broadly depleted, really dramatically depleted by a period of early life stress. However, that binding recovers completely to normal in adulthood. In fact, there's even evidence of some overshoot, even though that was not statistically significant. Now, we're in the process now of doing this experiment genome-wide by chip sequencing for OTX2, although it will require improvement in the antibodies that are currently available. So the question is, how is it that this transient reduction in OTX2 at a gene is causing essentially a permanent, what we call chromatin scar at the gene, so that even though OTX2 binding recovers to normal after a few days, the expression potential of that gene remains suppressed or altered for a lifetime? We're now approaching this in several ways. One way depicted here in collaboration with Ian Mays at Mount Sinai and Ben Garcia's lab at Penn. We're using an open-ended proteomic analysis to identify what are the changes in chromatin genome-wide that are associated with early life stress and OTX2 depletion. Again, emphasizing the importance of open-ended, unbiased analyses, not simply picking one out of 100 histone modifications and seeing whether that might be involved. So this proteomic analysis identified 3 chromatin marks that are depicted here. I'm just going to give you a quick code for what this represents. Monomethyl lysine 4 of histone H3, or trimethylation of the same mark, or dimethylation of lysine 79 of histone H3. It does not matter what that means for those of you who are not chromatin biologists. These are simply types of biochemical modifications of histone proteins at specific amino acid residues that are related to gene expression in diverse cell types. These were the 3 that are changed. Therefore, these are the 3 for which we are performing chip sequencing, and I'm showing you chip sequencing data for one of these marks, the monomethyl lysine 4 mark. The ChIP-seq data validated the proteomic analysis that there is indeed a genome-wide enrichment of this chromatin change. This is looking at all genes averaged together, focused at the transcription start site of these genes, then looking at 3,000 bases up and downstream of those transcription start sites. And you can see an appreciable enrichment of this mark after early life stress. This is simply one of the target genes where one can take what's called a genome browser, enlarge it, and show where across that gene this increased binding is occurring. We are now taking these ChIP-seq runs on these established chromatin changes that occur after early life stress and comparing them to our other data sets. One of the comparisons is shown in the heat map on this slide. This is showing all the genes that show a significant increase in this one histone mark after early life stress. In general, one can see that most of these genes are actually suppressed at the mRNA level under baseline conditions after exposure to early life stress, although some are induced, but broad enrichment of the same genes in both cases. And even the genes that seem to be suppressed after early life stress, those in blue, seem to be induced, turn to yellow, in animals exposed to 2 hits, early life stress plus a subsequent adult stress. So we think that these are very encouraging preliminary data. We'll need to examine the other chromatin marks to establish the nature of these chromatin scars. What is OTX2 doing to these target genes to affect VTA function? I'm just going to run through a few slides and then wrap up. Remember I mentioned that ion channels are prominent among the predicted gene targets of early life stress and OTX2, and accordingly, in collaboration with Ming-Hu Han's lab at Mount Sinai, Kate and others have been able to show that early life stress does indeed cause an increase in the electrical excitability of VTA dopamine neurons. This seems to be recapitulated by a loss of OTX2 using the viral vector that knocks down OTX2, suggesting that transient suppression of OTX2 might cause a lifelong change in the excitability of VTA dopamine neurons. And this observation jives very nicely with work from Ming-Hu Han's lab and my lab, several papers where we've been able to show that an increase in the electrical excitability of these VTA dopamine neurons is a mechanism of enhanced stress susceptibility. This seems to be correlated with alterations in the projections of VTA dopamine neurons, showing that after early life stress, there seems to be an increase in dopaminergic innervation of the nucleus accumbens, a trend toward a reduction in dopaminergic innervation of the prefrontal cortex. Again, this is an effect only seen in male mice, not in female mice. And what about female mice? What about when we drill into the data sets of female mice, we see very strong upstream regulators there as well, just different from what's occurring in the male mouse brain. So as just one example, here's a heat map showing the same type of concordance between the effects of early life stress and the effects of a second hit of stress in the female VTA. The signal is just as strong as in the male, only the upstream regulators are almost completely different. And the gene that shows the strongest predictive value for these concordantly regulated genes is BDNF. So we know that BDNF has long been studied by my lab and many others as controlling stress responses in male mice. These data suggest an important role in females as well. So with that, let me just conclude. We think that this early life stress paradigm and others like it are very interesting from a heuristic point of view in allowing us to address the question of how life experience early in life can program an individual's susceptibility for stress for a lifetime. I've demonstrated that although this occurs in males and females to an equal extent, the underlying molecular mechanisms seem to be mostly different between the sexes. In the male VTA, OTX2 is important. A transient reduction in OTX2 triggers lifelong impairments in its target genes, even though OTX2 returns to normal, really driving us to understand the molecular basis of those lifelong chromatin scars that are responsible for this programming. And then doing the same types of analyses in the other brain regions in both sexes. So with that, let me conclude. I hope I've convinced you that it's possible to learn a great deal about stress responses, but also about human depression by studies of mouse models. I focused today on sex differences and on lifelong changes in susceptibility and resilience caused by early life experience. Other focuses in the lab are looking at mechanisms of antidepressant action with an interest in identifying novel mechanisms. We believe that studies of chromatin are important in helping us understand how life experience changes our behavior for a lifetime. And of course, the challenge is to use this information to develop new diagnostic tests and treatments for this very, very broad syndrome that we call depression. I have to say, I've had a lab now for 30 years. We thought 30 years ago that these types of approaches would lead to dramatic improvements in the clinic within 5 or 10 years. We have not seen that. So it's with a great deal of humility that I'm here. On the other hand, when I was in medical school close to 40 years ago, we were taught that the era of molecular medicine was with us and that it would be possible to design individualized treatments for cancer and other diseases. And it's taken us 40 years, 4 decades, to finally get to the point where we have the first precision medicine approaches for cancer, cardiovascular disease, immunologic disease, and so on. The brain is far more complicated. The diseases of the brain are far more complicated than cancer. And I just think the slow pace of progress reflects that complexity. But I remain as optimistic as ever, and it's a challenge to the young people in the audience to harness the truly transformational tools that we now have in neuroscience at the molecular level, which I focused on today, but also at the circuit level, and you'll hear more about that from Kaf Jarasa later this morning. Merging those two together, I think, will perhaps get us to where we need to be. So with that, I will show you my hometown. Welcome to New York. And I'd be happy to, I think we do have a couple of minutes for questions. Thank you.